CRISPR, clustered regularly interspaced short palindromic repeats, is a revolutionary gene editing technology that allows scientists to precisely modify DNA in living organisms. Originally discovered as part of a bacterial immune system, CRISPR has been adapted to edit genes by using an enzyme called Cas9, which acts like molecular scissors to cut DNA at specific locations. This technology has the potential to cure genetic diseases, such as sickle cell anemia, cystic fibrosis, and even some cancers, by targeting and repairing faulty genes. Researchers are also exploring CRISPR's ability to enhance agricultural crops, making them more resistant to pests and environmental stress, and to combat viruses by editing viral DNA. One of the most exciting possibilities is in the field of germline editing, where CRISPR could be used to edit embryos, potentially eradicating genetic disorders before a person is even born. However, this raises significant ethical concerns about designer babies, unintended consequences, and the potential for misuse. While CRISPR has shown incredible promise, it's still in its early stages, and further research is needed to ensure its safety and accuracy. If fully realized, CRISPR could reshape medicine, agriculture, and our understanding of genetics.